Hello everyone, my name is Hebah Elissa, and today I will be presenting about the article Expanding the Human Gut Microbiome Atlas of Africa. So out of all the microbiomes in our body, the human gut microbiome is by far the largest, most dense, and diverse microbiome in the human body. The gut microbiome plays a huge role in metabolic rate, digestion, disease development, behavior and mood via the gut-brain axis, and protecting against pathogens. Sadly, most of the research in this area has been conducted on high-income countries, and that has led to a huge gap in research between low-middle income countries and high-income countries. And the main focus of this research study was to help expand the available gut microbiome data of Africa and help close that research gap. The main question that the authors aimed to answer was what geography, lifestyle, and disease factors are responsible for the microbial diversity observed in various underrepresented African populations. This study was hugely successful. In fact, it is the largest African population gut metagenome study to date, and it discovered novel microbial genomes that we had not identified before and were not in any kind of database. 1,000 new bacterial genomes were identified and over 40,000 new viral genomes, including 9 novel jumbophages. It also linked HIV with decreased microbial diversity, and it was a great model for ethical research practices with diverse populations. The field of gut microbiome research is rapidly growing, and its importance is becoming more globally recognized. In fact, scientists have begun referring to the human microbiome as our second genome because of the various functions the microbiome plays in our bodies. In 2007, the Human Microbiome Project was launched by the National Institute of Health. This project established the core human microbiome, which served as a starting point for further research efforts. As with most of the other existing literature, this study was geographically narrow, as it oversampled individuals from high-income countries such as the US and Western Europe. Since these populations have similar diets, lifestyles, environmental exposures, resource access, and disease profiles, it hinders our ability to generalize these studies' findings to the global population. The current research severely underrepresents low and middle income countries in gut microbiome research, and large microbiome studies rely on facility-based convenience sampling. This sampling method is based on availability and can result in convenience bias and low external validity. So why do we need this study, and what's the urgency of the situation? Low and middle income countries host 84% of the global population. To put this percentage into perspective, approximately 6.8 billion people do not live in high-income countries, yet most of the gut microbiome research is based off of individuals in such countries. There's also a pressing need for more studies to utilize a random cross-sectional population sampling, as this research study did, as most of our current literature focuses on using facility-based convenience sampling. By using cross-sectional population sampling, we can increase generalizability, there's going to be less selection bias, more external validity, and more accurate representation of population diversity. It is important to note that many research studies opt to use facility-based convenience sampling as it is less resource intensive. So what is the broader implication of this? Well, we are going to be able to have a more equitable global health system. There's going to be an increased ability to develop precision medicine and formulate medicine and microbiome-based therapies based on the distinct and unique African population gut microbiomes. There's going to be more inclusive reference database, and we can actually rethink what a normal gut microbiome is, as normal varies based on where you are on the globe and what region you're in. The main research objective was to map and analyze the gut microbiome of individuals in various diverse African communities in order to build a better reference database. The three main goals that the authors set out to achieve was to understand the microbial diversity of the gut microbiome across geographic and lifestyle borders. They wanted to discover novel microbial species, and they wanted to link certain gut microbiome alteration with HIV outcomes, so whether someone is HIV positive, HIV negative. On to methodology. The study's design was a cross-sectional gut microbiome study using the AWI Gen 2 framework. There were 1,820 adult participants across varying locations in South Africa, Ghana, Kenya, and Burkina Faso, and there were 1,801 women and 19 men, and the men were largely excluded from the analysis because of the small sample size, which led to lower alpha diversity, and that could have led to potential bias. And the study's downstream goal was to pair with a menopause-related study, so including those men would have inhibited that. Exclusion criteria: pregnant women, people in the same household, and individuals with less than 10 years of residency. Sampling method: random cross-sectional population sampling. The sample collection was DNA extraction from stool samples. And the sequencing method was shotgun metagenomic sequencing. And this analyzes all of the DNA in a sample, regardless of whether it is a virus, bacteria, archaea, or fungi. This means it can capture multiple domains of life rather than being limited to bacteria, as in 16S RNA sequencing, for example. The platform used was Illumina sequencing with paired and 2x150 base pair reads. And Illumina sequencing results in short reads, which makes it harder to assemble complete genomes. It also has a high throughput, which makes it ideal for the study at hand. And the output was 44.16 million median reads per sample, and the usable ones were 31.2 million. So I've included this shotgun metagenomic illustration to help visualize the process. So here you can see the environmental samples. In our case, we used stool samples, and then the DNA extraction was conducted, and then the DNA was sequenced and analyzed. Genomes were assembled and then compared to databases in order to discover if they were novel or not. There were multiple statistical approaches used here in this study, so I've just outlined a few of the main ones. Inverse Simpson's index for alpha diversity and Bray-Curtis distance for beta diversity, principal coordinate analysis, Kruskal-Wallis test, Wilcoxon test, ANOVA, and distance-based redundancy analysis. And there's also two main terms I'd like to just define briefly. Metagenome assembled genomes are microbial genomes that are constructed from the DNA sequences that we extracted from environmental samples, so in our case, the stool samples. And then jumbophages are essentially just larger than normal genome bacteriophages, so they have over 200 kilobases in their genome. I've included a few existing and possible confounding variables such as site, diet, migration, lifestyle, antibiotic use, probiotic use, diarrhea. I'd focus on genetics just for a second. The role genetics play in shaping the gut microbiome is considered to be less influential than the role of the environment, diet, lifestyle. However, this doesn't mean that genetics is not worth investigating. Booland and al. 2022 study was a research study that found ethnicity-specific genetic associations with gut microbes in the human body, which I think is interesting and worth further exploration down the road. A few others would be sanitation and socioeconomic status. All right, on to our first figure, figure 1a. So this figure is essentially just a background that helps us contextualize the number of participants in the study from each location and the nature of the geographical regions that the samples were drawn from. For instance, we see that Nanero and Navrongo are horticultural regions, which prioritize the growing of plants for sustenance. Meanwhile, Dimamo and Agincourt are semi-rural, meaning it's an intermediate between rural and urban. Soweto is a post-industrial region, which focuses on services. Lastly, Nairobi is an industrial region, which means it is dominated by business and trade and is the most urbanized of the other sites. This figure is important to discuss and look through in order to understand the geographic locations of the regions discussed in the paper, as well as to understand the degree of industrialization each region possesses. And as we can see, the sample sites are geographically diverse and lifestyle factors are also diverse. On to figure 1c, the main takeaway from this is that prokaryotic diversity is higher in rural regions. So prokaryotic diversity was highest in Nanero and Navrongo, which are horticultural or rural regions. It decreased as industrialization increased. So along the industrialization axis, prokaryotic diversity decreased. The more industrialized or urban the country was, the less prokaryotic diversity identified. Interestingly, though, Nairobi, which is the most urbanized, industrialized of all the regions sampled, was not the lowest. Soweto, which is a district within Johannesburg, South Africa, had the lowest prokaryotic diversity. This is quite shocking, as one would expect Nairobi to be the least diverse in prokaryotic species. There are multiple reasons behind why Nairobi is an exception from the industrialization axis, including diet and migration. On to figure 1e, the main takeaway is that the abundance of taxa varies across sites. So we can see that the abundance of Treponema is highest in Nanaro and Navrongo. However, in the other more industrialized study sites, we can see that there's almost no Treponema present. This indicates that Treponema is a bacterium that is lost with urbanization, and it may be sensitive to antibiotics. A similar pattern is observed in Prevotella, Cryptobacterioides, and Oribacterium. On the other hand, Phocacola is more abundant in industrialized regions such as Soweto and less prevalent in rural areas such as Nanero. This indicates that the bacteria may thrive better with a fatty diet, or perhaps it's more resistant to antibiotics. This same pattern is seen in Bacterioides and Bifidobacterium. On to figure 2a. The main takeaway from this figure is that there are over 1,000 novel bacterial genomes discovered. So this figure here is a phylogenetic tree of over 2,500 MAGs. Over 1,000 novel genomes were discovered across 19 different bacterial phyla, as indicated by the inner ring. This means that we have been overlooking a significant portion of the African genome in our current literature, and it goes to show how groundbreaking this research study truly is, as it enlightened us. Now, looking at figure 2b, this figure is essentially just showing the novel bacterial MAGs and their associated phylum. So in this figure, we can see what proportion of the novel MAGs belong to each bacterial phylum. By observing the figure, we can see that the majority of the new bacterial MAGs belong to the phyla Bacillota A, Actinomycota, and Bacillota. Such phyla are involved in the fermentation of fiber and production of short-chain fatty acids, which are not abundant functions in previous microbiome studies that were focused on the Western world. This figure helps illustrate the unique function and composition of the African gut microbiome, and that has been lacking from our research literature and scientific understanding. Now looking at figure 2c, This is a figure that shows the novel bacterial genomes compared to UHGG database. So in this figure, we compare the proportion of bacterial MAGs already present in the unified human gastrointestinal genome, the UHGG catalog, with the novel bacterial MAGs. We can see that all the sites from rural Ninero to industrial Nairobi had novel bacterial MAGs, which the UHGG catalog failed to recognize. This is reflective of the gut microbiome data gap in African populations that the study aimed to help close. The novel bacterial MAGs that were discovered could be crucial in helping develop personalized microbiome-based treatments for African populations. So looking at figure 2e, the main takeaway from this is that Treponema circumfaciens abundance decreases with antibiotic usage. As we can see in the figure, Treponema S was much less prevalent in individuals who had more recently taken antibiotics. This means that modern medicine and industrialization are decreasing the abundance of Treponema S, which leads to the loss of ancestral gut microbes and the alteration of traditional gut ecology. In fact, the article states that in regions with a high prevalence of Treponema S, such as Nanaro, Burkina Faso, and Navrongo, individuals in that region utilize antibiotics less recently. So this bacterium is found in many rural and non-industrialized populations, but it is lost in industrialized regions such as the West, which do not only prescribe antibiotics, but perhaps most likely they overprescribe antibiotics, which is destroying our ancestral gut microbiomes. Looking at figure 3b, the figure compares the viral catalog sampled from the participants of the study with the metagenomic gut virus catalog, MGV. Even though the MGV catalog contains over 50,000 viruses, which is quite an impressive amount, the sampled viral catalog found over 40,000 new viruses. This means that the MGV was missing at least over 40,000 viruses, and probably much more, as efforts are made to continue and sample more and more diverse populations in order to get more meaningful and universal databases. Looking back at the figure, all the sites, whether rural or urban, contained novel viral genomes that made up the overwhelming majority of the viral genomes, when compared to the viral genomes that were identified in the MGV catalog. This figure is similar to figure 2c that compared the bacterial MAGs with the UHGG catalog that we looked at earlier. Figure 3b suggests that the viral gut genome is under-researched, especially with respect to unique viral genomes that are present in lower-income countries. Therefore, we must invest in more research efforts to diversify the viral gut genome database as well as the bacterial gut genome database. Looking at figure 3e, this figure is representative of genome maps of the 9 novel jumbophages, which are essentially phages with a large genome exceeding 200 kilobases that are not included in the MGV database. These jumbophages are widespread, and genetic aspects such as CRISPR arrays, which are short repeating DNA sequences with spacers that help microbes remember phages. Interestingly, multiple phages had unidirectional gene organization, which is not common as it means that almost all the genes are transcribed from the same singular DNA strands. This finding demonstrates how little we currently know about the viral genome in the African population's gut microbiome and how much research is needed to fill in our knowledge in this area. Now we'll be focused on linking HIV with gut microbial changes. So the main takeaway from this figure, 4b, is that the HIV results in reduced microbial diversity. So we consistently see across all the sites that the prokaryotic diversity of individuals who are HIV negative is greater than the prokaryotic diversity of individuals who have HIV. This suggests that HIV reduces the prokaryotic diversity of the human gut microbiome. This indicates that individuals with HIV have microbiome dysbiosis, which decreases the health of the gut microbiome and makes them more susceptible to other diseases. HIV patients often deal with chronic inflammation and immunodeficiency. In fact, based on a 2013 study by Okoye and Picker, HIV patients have significantly lower levels of CD4+ memory T cells, especially in areas such as the gut, which is our focus here. Therefore, it is vital to further investigate the relationship between HIV and the gut in order to better understand the disease itself and to formulate better microbiome-based therapies. All right, on to the last figure of the results section. The main takeaway is that there is a varying gut microbial abundance in HIV patients. So what you have in front of you is a heat map. On the left-hand side is Agincourt, in the middle is Soweto, and on the right-hand side is Nairobi. The red bars on top reflect the bacteria that increase in abundance in HIV patients, one of which is Fucibacterium mortiferum, and this is a non-inflammatory microbe. Of more interest, however, are the blue bars on the bottom, which reflect the bacteria that decrease in abundance in HIV patients, many of which belong to Faecalibacterium, which makes sense as this is a microbe known to reduce inflammation in the human body. Since we know that HIV leads to chronic inflammation in the body, it is logical that the Faecalibacterium abundance is decreased as it works to fight inflammation in the body. The other bacteria listed in the blue have not been contextualized in the gut microbiome field yet, but they have been recognized in environmental studies, so further research on these bacteria is necessary. The AWI Gen 2 Microbiome Project has expanded the available gut microbiome data of African populations tremendously and helped close the research gap between low and middle income countries, which have often been neglected in such studies, and high income countries, which are often the focus of these studies. We learned that African populations have distinct gut microbial compositions, and we already know that they are severely underrepresented in the literature, but now we know the extent of that underrepresentation and the negative consequences that it has. We know there are over 1,000 novel bacterial species and 40,000 viral genomes that are novel as well. We also have linked HIV with certain gut microbiome changes. There are various implications of this study, one of which is the possibility of personalized medicine, aka precision medicine. Now that we know the distinct and unique African population's gut microbiome, we can better formulate medicine for their microbiomes, and this leads to a more equitable global healthcare system. We also have a more global representation of gut microbiomes with the new information from the study, and more equitable research practices as the study was ethically designed. We still do not know a couple of things, one of which is the impact of diet. That was not really considered in the study. We also do not know the longitudinal microbiome changes, as this was a cross-sectional study, so perhaps future research could focus on conducting a longitudinal study. We don't know the connection between the genetics of the host and the environment, So that is always a future research endeavor. And we don't know what causes the microbiome changes. The HIV does HIV cause the associated microbiome changes, or do the microbiome changes occur prior to HIV? To sum up, this cross-sectional study has transcended cultural barriers and expanded our knowledge of the gut microbiome, composition, and diversity of African populations. With our new knowledge, we must continue striving for greater inclusion of low and middle income countries. We shouldn't settle for just one or two research studies. We want as much literature as there is for the Western population in order to get a more equitable society. Here I've listed 5 questions for the discussion. 1, should we develop regional healthcare, healthy microbiome databases instead of a universally standardized definition, and what are the advantages and disadvantages of doing so? 2, now that we have discovered novel bacterial MAGs and viral genomes, should we begin to shift into attempting to discover their functions? If so, what would be the best approach? 3, we know that the more industrialized a region is, the less gut microbial diversity its residents possess. What do you think is the main factor of industrialization that drives this reduction in microbial richness? 4, as discussed in the presentation, HIV infection decreases the prokaryotic diversity of the gut microbiota. Which mechanisms do you think are behind this, and why would it be worthwhile to investigate them? 5, if you were asked to design a follow-up study, how would you go about designing it, and what would your research focus on beyond? This is my reference slide, and thank you all so much for watching, and I hope you gained something from this presentation and found it as interesting as I did.